Welcome to this tutorial on the visual system, and specifically I'd like to talk to you in this session on central visual processing. We're going to talk about some of the most wondrous and most interesting aspects of cortical physiology that will highlight once again the complexity of the brain among the body's organs. We will encounter some circuitry that's genetically determined, and that circuitry will form the foundation for visual processing in the brain. And some of the topics we will consider today are just things that just fill me with a sense of awe and wonder at what the human brain can accomplish. And that sense of wonderment is what drives my own personal curiosity to try to understand how the brain works. I have several learning objectives for you today. I want you to be able to describe the distribution of the axons of retinal ganglion cells to the major processing centers in the forebrain and the brainstem. And then I want to focus on what's going on at the level of the forebrain, specifically in the primary visual cortex. I want you to be able to discuss the major receptive field properties that we discover when we record electrical signals from neurons in V1. I want you to be able to discuss in very general terms the functions associated with those parts of the visual cortex that are beyond the primary area. We call those extrastriate visual pathways. All right, let's once again consider the distribution of the axons of retinal ganglion cells. So the ganglion cells of the nasal retina send their axons through the optic nerve. They cross the midline and project to targets in the diencephalon and in the midbrain on the opposite side of the brain. Those retinal ganglion cells from the temporal retina send axons that grow through the optic nerve, and as they approach the chiasm, they remain on the ipsilateral side of the brain. And as a consequence, once we get visual signals posterior to the optic chiasm, now each optic tract contains all the information about the opposite side of the visual world that's seen by each eye. Now, the major target of this visual information with respect to the image-forming aspects of vision is the lateral geniculate nucleus. And we see that nucleus illustrated here in the posterior and inferior and lateral aspect of the thalamus. It's the major target of retinal ganglion cells with respect to vision or image formation. And from the lateral geniculate nucleus arises our optic radiations that work their way around the lateral ventricle to supply the upper and lower banks of the calcarine sulcus. That's where we find our primary visual cortex. Now, here to the right is a histological view of the primary visual cortex that was generated by a good friend and colleague of mine back in the day when we were both working as postdoctoral fellows together. And this is a beautiful histological view of a myelin stain through the cortex that forms the calcarine sulcus. So here in this space, here's the calcarine sulcus as labeled there. And you'll notice if we look into the thickness of the cortex with the myelin stain that there's this striking dense band of myelination that runs right through the middle of the cortex. And one feature of this band that is especially striking is how it so clearly defines a region of cortex here. One can recognize exactly where this band has its terminations. And the early anatomists, such as Corbity and Brodmann, looked at this part of the cortex and concluded that the presence of this stripe, which is called the stria of Gennari, must signify that there's something distinct about this bit of the cerebral cortex. So Brodmann labeled this Brodmann's area 17, or that's how we know it today. He just called it area 17. And it's also known as the striate cortex because of this histological presentation of what we call the stria of Gennari. Now, one interesting aspect in the history of neuroscience over the last 5 or 6 decades or so is that probably no single area of the brain has attracted more interest, more attention than this striate cortex. And one reason is that it's within the circuitry of the striate cortex that we come best to understand this computational function of cortical microcircuits. You will recall, I trust, what I suggested is an easy way to remember the function of the cortical microcircuitry, the ACC of the cortical column. The A stands for amplification of the thalamic input. The C, the first C, stands for computation. That is the emergence of new functional properties that weren't present in the input signal that is amplified by this cortical circuitry. We'll see some wonderful examples of computational properties that emerge as a result of the operation of cortical networks. And then the final C is communication, as these signals are then disseminated along higher order pathways. We'll see an example of that here as well. So the ACC of the canonical cortical microcircuit was truly first established by studies of this part of the brain, the visual cortex. Two of the leading investigators that has made all of this possible are David Hubel and Torsten Wiesel, who very deservingly were awarded the Nobel Prize in Physiology or Medicine in 1981 for their foundational work on the physiology, the anatomy, and the developmental plasticity of this part of the brain. It's really difficult to overstate the influence of these two scientists on the development of modern neuroscience. There were others at the time that were beginning to eavesdrop on the nature of neural signals in the cerebral cortex. Most notably at that time was Vernon Mountcastle, who was studying cortical columns in the somatic sensory parts of the brain. But for the visual system, it's really David Hubel and Torsten Wiesel that paved the way for generations of neuroscientists who would follow, including myself, who have been fascinated with trying to understand the physiology of the circuits that arise in this part of the brain and how they might be shaped by our visual experiences in early life as we open our eyes and begin to encounter the world. Well, I could go on about Hubel and Wiesel and their influence over my career and the career of many of my colleagues, but we will press on and turn our attention to some of the receptive field properties that Hubel and Wiesel discovered in this part of the brain. And I'd like to share a little bit from my own research lab, some of the ways that we are continuing to build upon what Hubel and Wiesel taught us and hopefully extend our understanding of this fascinating part of the brain. Now, as we get into this discussion, let me remind you that at earlier stages of visual processing, such as in the retina and even in the lateral geniculate nucleus, cells respond best to small spots of light. They have receptive fields that are organized into center and surrounding regions that are circularly symmetrical. And so what we see in the thalamus is very much similar to what we saw in the retina. There's no major transformation of the nature of visual representation from retina to thalamus. However, if we record from cells in the visual cortex and try to stimulate with the same stimuli that works so well for retinal ganglion cells or lateral geniculate nucleus neurons, we see very subtle and not terribly robust responses. But rather, if we change the stimulus, if we replace the small spots of light with large edges of light or bars of light, and then move those bars in one direction or another at one orientation or another, let's say horizontal or vertical, then we are very likely to record robust responses in the primary visual cortex. And this is exactly what Hubel and Wiesel did. And what they discovered are two very important computational properties of this cortical network: orientation selectivity and direction selectivity. Let me illustrate what they found by considering cortical orientation selectivity. So Hubel and Wiesel did experiments such as this using animal models, and this highlights the importance of animal models in modern neuroscience. Of course, we are highly concerned for the welfare of all animals that are used in science or those animals that are kept as pets by private individuals as well. We need to treat these animals with the highest standards of humane concern for their welfare, and that's certainly the case, generally speaking, in the field of neuroscience. I know in my own work with animals and research, the standard is to keep these animals as comfortable and as healthy as we possibly can. Keep the animals socially engaged, supported in their nutrition and in their physical activity. And in the conduct of the experiments, the goal is to eliminate the experience of pain. So consequently, when experiments are done on animals, they are anesthetized before any intervention is done. And while the experiments are ongoing, the animals are very carefully monitored to be certain that they are appropriately anesthetized in order to ensure that there is no pain experienced by the animal. And in the case of terminal experiments, at the conclusion of the study, the animal is simply put down with an overdose of anesthetic. Well, this is the kind of experiment that was done by Hubel and Wiesel using cats and young kittens. And an illustration of one such experiment is shown here. We have an animal that's been anesthetized and prepared for surgery, and it's anesthetized to the point where it's feeling no pain, but not so deeply that there's no brain activity at all. Otherwise, of course, that would be incompatible with life. So the animal is alive, but it's deeply anesthetized, much like a person undergoing surgery. Well, a microelectrode is implanted in the visual part of the brain of this animal, back in the occipital cortex. And from that microelectrode, action potentials are recorded from neurons while these animals view light stimuli that are projected on a screen in front of the animal. As I mentioned, what Hubel and Wiesel discovered is that cells in this part of the brain respond especially well to a bar of light when it's moving in a particular axis of motion. And what they might even find is that some neurons respond very well when the bar is moved in one direction, but not at all when the bar is moved in the opposite direction. So these two properties together are what we refer to when we talk about orientation and direction selectivity. Orientation selectivity refers to the preferred response to a bar of a particular orientation versus another, and direction selectivity refers to the preferential response in a particular direction of motion, but not in the opposite direction. Okay, so here in the middle panel, we see a representation of data recorded from one such experiment, and the microelectrode tip happens to be sitting next to a cell that responds best to vertical stimuli. So when the preferred stimulus is presented, there's a robust discharge of action potentials from that cell. But as the orientation or as the angle of that stimulus is shifted away from vertical, the response gets less and less. After a series of such stimuli that are presented around the full cycle of possible orientations, it's possible to define a tuning function that plots the response of the neuron across a range of stimulus presentations. And for this neuron, what we find is that there is sharp tuning for angles near vertical. Now, there's not absolute tuning. There is some response off vertical, but by far the best response is within about 15 or so degrees of the preferred orientation, vertical in this case.